Welcome to another deep dive. Today, we are unpacking something that is, frankly, one of the most notoriously stubborn puzzles in modern medicine. We're talking about pancreatic ductal adenocarcinoma, or PDAC.
Yeah, PDAC. And, when you look at the survival statistics for this specific cancer, the numbers are just incredibly grim.
Right. I mean, we're talking about a disease where less than 5% of patients see any kind of meaningful response to current immunotherapies. 5%.
Yeah, exactly. You can literally flood a patient's system with the most advanced genetically engineered immune cells that modern medicine has to offer. And for 95% of people, those therapies just bounce right off.
It's an incredibly aggressive, stubborn disease, which I think is what makes the anomaly so fascinating, because in a very small subset of human patients, the immune system actually manages to breach the tumor.
Right. It doesn't just infiltrate. It sets up a permanent camp. And we call these structures tertiary lymphoid structures, or TLS.
I love the concept of a TLS. I always like to think of them as these unauthorized, pop-up military bases.
That's a great way to put it, actually.
Because normally, your immune system's T cells and B cells are just hanging out in your natural lymph nodes, right?
Yeah.
Waiting to be deployed to an infection. But a TLS is like a rebel outpost built right inside the hostile territory of the tumor itself.
Yeah, complete with its own supply lines, its own specialized compartments for B cells and T cells. It's actively directing traffic. And patients who naturally develop these pop-up bases inside their pancreatic tumors, they survive significantly longer.
They have a massive survival advantage, right? Because the immune system is basically fighting the war from inside the castle walls.
Exactly. And that brings us to the source material for today. We are pulling from a really groundbreaking 2026 paper published in the journal Cancer Cell.
Right.
And this paper set out to answer the obvious question, why do some pancreatic tumors allow these immune outposts to form while others remain completely impenetrable?
And I guess the secondary, much bigger question for anyone listening is, can we artificially force a stubborn tumor to build one of these bases?
Yes. And the researchers took a brilliant angle here to figure that out. Instead of agonizing over the immune cells themselves, which is what we usually do, they shifted their focus to the tumor's real estate, the physical scaffolding.
Okay, so let's get into the mechanics of that, because they started with genetically identical cancer cells, right?
Right. They derived these cells from the exact same lineage of genetically engineered mice, specifically KPC mouse models, which are the gold standard for pancreatic cancer research. And they implanted these identical cancer cells into new hosts.
So on paper, you've got twin tumors, same driver mutation, same starting point.
Exactly. But in reality, once implanted, they behaved completely differently. Some of these tumors naturally allowed immune infiltration. The researchers termed these TLS-permissive.
Okay, permissive tumors.
Yeah, and others formed an absolute fortress, completely blocking the immune system, and those were categorized as TLS-resistant.
So you have two genetically identical seeds, but they grow into completely different environments. And the researchers wanted to see if they could manually trigger the permissive tumors to build an outpost. Right. So they introduced a specific drug, an LTBR agonist. Can you unpack what that is?
Sure. So LTBR stands for lymphotoxin beta receptor. During embryonic development, when you're just a fetus, your body uses the LTBR genetic pathway to grow your natural lymph nodes.
Oh, wow. So it's the fundamental building block pathway.
Exactly. By using an agonist, which is a compound designed to artificially stimulate that exact receptor, the researchers were essentially sounding a chemical alarm, tricking the tissue into thinking, hey, it's time to build lymphoid architecture right here, right now.
So they hit the build button. What happened?
In the TLS-permissive tumors, hitting that LTBR switch worked beautifully. B cells and T cells immediately flooded the area. They aggregated into these early-stage pop-up bases, and the tumor shrank.
That's incredible.
And here's the catch. When they applied that exact same chemical alarm to the TLS-resistant tumors, absolutely nothing happened.
Wait, really? Nothing?
Nothing. The signal was completely ignored, and the tumor just kept growing.
But wait, if the switch is functionally the same and the genetics of the cancer cells are identical, why is this signal being ignored by the resistant tumor right next door? What is muting the alarm?
Right, that is the million-dollar question. And to answer it, we have to look past the malignant cancer cells themselves and examine the tumor microenvironment, specifically the fibroblasts.
Okay, fibroblasts, let's define those for the listeners.
So fibroblasts are the cells responsible for synthesizing the extracellular matrix. They lay down collagen, they create the structural framework for our tissues.
They are the construction crew.
Exactly. But in a tumor, They get hijacked and become cancer-associated fibroblasts, or CSFs.
Okay, so the tumor hijacks the construction crew, and the paper reveals that the construction crews in the permissive tumors look entirely different from the crews in the resistant tumors, right?
Night and day difference. The permissive tumors are full of what the researchers call reticular myCAFs, or rCAFs.
The builders. Or, actually, I like to think of them as city planners.
City planners is a perfect analogy. These rCAFs are highly specialized for organization. They express surface proteins like ICAM1 and VCM1.
And those are basically molecular Velcro, right?
Yeah, molecular Velcro is a great way to visualize it. When an immune cell is floating by in the bloodstream, these proteins snag it. They allow it to stick to the tissue so it can cross over into the tumor.
But snagging them isn't enough. You have to give them directions once they're inside.
Right. So these rCAF city planners also secrete very specific chemical breadcrumbs. These are chemokines known as CXCL13 and CCL19.
Let me guess. One is for B cells and one is for T cells.
You got it. B cells are naturally drawn to CXCL13, and T cells follow the CCL19 scent trail. The rCAFs are literally paving the roads and laying down a scent track to guide the immune system into the core of the tumor.
That is so elegant. But contrast that with the construction crew found in the TLS-resistant tumors.
Yeah.
Because those are the ones where the immunotherapy fails.
Yeah. Those tumors are utterly dominated by a different type of fibroblast. Myofibroblastic CFs or myCAFs. These are the bad guys. The blockers.
Exactly. These cells do not express the molecular Velcro. They do not secrete the chemical breadcrumbs. What they do is express massive amounts of a protein called alpha SMA, which stands for smooth muscle actin.
Smooth muscle actin. So it makes them like contractile.
Right. It gives these cells a contractile, almost muscle-like property. They pull the tissue tight.
So if the rCAFs are the organizers laying down roads and setting up beacons, the myCAFs are like hostile contractors pouring thick, impenetrable concrete over the entire site.
That's exactly what it looks like under a microscope. It creates a condition known as the desmoplastic stroma. It's this dense, highly pressurized, fibrotic scar tissue.
Wow.
The physical pressure inside a desmoplastic tumor is often so high that it literally collapses the surrounding blood vessels. The immune cells couldn't get in even if they wanted to because the roads are physically crushed.
Okay, so that structural density explains the physical barrier. But the paper found something much more insidious, didn't it? The myCAFs aren't just physically blocking the immune cells. No, they are actively fighting a chemical turf war. They are running a chemical jamming signal to suppress the city planners.
And the villain molecule powering that jamming signal is a cytokine called TGF-beta.
Yeah, TGF-beta. It is one of the most potent immunosuppressive forces in the body's entire arsenal, and the researchers mapped out exactly how TGF-beta maintains this fortress state using a series of highly controlled in vitro experiments.
So they isolated the cells in a lab dish to watch the mechanics unfold. Let's walk through that because it's fascinating.
Yeah, so they took naive, unprogrammed fibroblasts, just blank slate cells, and they exposed them to the node-building signal we talked about earlier, the LTBR agonist.
Along with another molecule called TNF-alpha, right, which acts as like an inflammatory amplifier.
Right, and when given those specific instructions, the naive fibroblasts transformed into the helpful rCAFs. They started pumping out the CXCL13 breadcrumbs. They successfully became city planners.
Okay, but what happens when you introduce the villain, when they added TGF-beta into the mix?
The outcome flipped entirely. If the naive fibroblasts were pre-exposed to TGF-beta, that jamming signal fundamentally altered their gene expression. It physically downregulated their receptors.
Wait, so it silenced them?
Completely. Even when the researchers subsequently flooded the dish with the LTBR build signals, the fibroblasts were deaf to it. They physically could not receive the order to become rCAFs. TGF-beta locked them into becoming the hostile myCAFs.
Wow. And the researchers demonstrated the real-world consequence of this with a migration assay, right? The transwell assay.
Yeah, it's an incredibly elegant setup to prove the concept.
I love this experiment. You have a microscopic well with two chambers separated by a porous membrane. You put the fibroblasts at the bottom, and you put a mix of T cells and B cells in the top chamber.
Right. It tests chemotaxis, which is just the ability of cells to move in response to a chemical stimulus.
So what happened?
Well, when the fibroblasts at the bottom were successfully acting as rCAFs, pumping out those chemical breadcrumbs, the immune cells up top immediately squeezed through the porous membrane and migrated down to the bottom chamber.
You followed the scent trail.
Exactly. But the moment TGF-beta was added to the fibroblasts below, the migration stopped completely.
Wow.
The B cells and T cells just sat in the upper chamber. The beacons had been silenced.
Which perfectly explains the 95% failure rate of immunotherapy in human pancreatic cancer, doesn't it?
It really does. I mean, we can engineer a patient's T cells to be the most lethal, precise weapons imaginable. But if those T cells are circulating in a patient whose tumor is dominated by myCAFs and flooded with TGF-beta, the weapons will never reach the battlefield.
Because there's no molecular Velcro to grab them, no chemical trail to guide them, and a literal wall of pressurized scar tissue keeping them out. Okay, so the mechanics of that are terrifying. But it also provides a really clear target. Let me challenge you here. If TGF-beta is the jamming signal actively maintaining this concrete wall, what happens if we cut the signal? Is this permanent? If a tumor is resistant, is it a lost cause, or can we deprogram these cells?
That is exactly where the paper moves from identifying the problem to testing a potential solution. A therapeutic flip.
Turning back the clock on resistant tumors.
Yes. The researchers designed a double-punch combination therapy to test on those highly stubborn TLS-resistant mouse tumors. The first punch is a TGF-beta receptor 1 inhibitor.
Okay, a drug designed to plug up the receptor so the jamming signal can't connect.
Right. And the second punch is the LTBR agonist, the drug that sounds the alarm to build the immune outpost.
So they're simultaneously cutting the jammer and blasting the villi.
And a local scaffolding actually responds. It's amazing. By blocking the TGF-beta receptors, the fibroblasts essentially wake up. They shed their hostile myCAF identity. And because the LTBR signal is suddenly coming through loud and clear, they repolarize.
The concrete wall cracks.
Yes. The cells transition back into rCAFs. They reboot. The repolarized fibroblasts immediately start transcribing the genes for CXCL13 and CCL19 again. They secrete the chemokines, and suddenly the immune rescue begins.
So the B cells and T cells rush into a tumor that was previously completely dead to the immune system, and they organize into those pop-up military bases.
Right. And they saw a massive reduction in tumor growth, particularly when this reprogramming therapy was combined with standard chemotherapy, because the drugs finally have a physical way in.
That is a massive breakthrough. But there is a crucial detail here about the immune rescue that we shouldn't skip. The dynamic between the B cells and the T cells within those newly formed structures.
Yeah, right. This is a fascinating nuance. The researchers noticed that the B cells are the primary architects of the pop-up base. They cluster together and physically form the aggregate.
Okay, they build the barracks.
Yeah, but when the researchers systematically depleted the T cells from the mice, leaving only the B cells, the therapy failed entirely.
Wow. So the B cells act as the engineers building the barracks, but they don't actually fight the tumor. The T cells are the actual soldiers doing the killing.
Exactly. If you build a pristine military base, but you have no soldiers to occupy it, the tumor just continues to grow. You absolutely need the fibroblasts to invite the B cells, you need the B cells to build the structure, and you need the T cells to execute the cancer cells. It's a full cascade.
And to prove that this entire cascade hinges on the fibroblasts, the researchers did something really clever with a mutant mouse model. They knocked out a gene called ALK5.
Right, ALK5.
And ALK5 is the gene that codes for the TGF-beta receptor itself, correct?
Correct. But here is the trick. They didn't knock it out of the whole mouse. They deleted the ALK5 gene only in the fibroblasts.
Oh, wow.
Yeah, so the cancer cells, the immune cells, the blood vessels, they all still had normal TGF-beta receptors. Only the structural fibroblasts were genetically deaf to the jamming signal.
So what happened when those specific mutant mice developed pancreatic tumors?
They naturally fought them off far more effectively. Because the fibroblasts never received the order to pour the concrete, they remain permissive.
That is the smoking gun. It proves beyond a shadow of a doubt that controlling the structural scaffolding dictates the entire fate of the tumor's immune response.
Oh.
The fibroblasts are the absolute linchpin.
Absolutely.
Okay, so mice are great and in vitro dishes are great, but I know anyone listening to this is wondering, does this apply to actual human patients?
Right. The necessary final step. To prove that this exact spatial relationship exists in human pancreatic cancer, the researchers utilized a cutting-edge technology called spatial transcriptomics.
I love this tech. Spatial transcriptomics sounds like absolute science fiction. Because, in traditional sequencing, you take a biopsy of a tumor, you basically grind the whole piece of tissue up and you sequence the genetic material.
Right. Bulk sequencing.
Yeah. You get a list of all the genes that were active, but you lose all the context of where those cells were actually located. It's like putting a completed jigsaw puzzle into a blender.
Yeah.
You know all the pieces are there, but the picture is destroyed.
Exactly. And spatial transcriptomics solves the blender problem.
How does it work?
You take a microscopically thin slice of intact human tumor tissue and you place it on a highly specialized slide. And this slide is covered in millions of microscopic barcodes.
Okay.
When the RNA transcripts from the tissue bind to the slide, they get tagged with a coordinate. So you can then sequence the tissue and map every single active gene back to its exact X and Y coordinate on the original slide.
So you end up with a high-definition molecular map of the actual battlefield.
Exactly. And the researchers applied this technology to actual tissue samples from human PDA patients who had naturally formed those rare tertiary lymphoid structures.
And what did they see?
They looked specifically for the gene signatures of the rCAFs, the builders, and the myCAFs, the blockers. And the visual data is just striking. The spatial maps showed the rCAFs physically clustered around the immune cells. They were forming a protective ring, literally hugging the B cells and T cells that made up the lymphoid structure.
Wow. They were maintaining the integrity of the base.
Right, and when you look for the myCAFs, the hostile blockers, they are completely banished from that area. The spatial maps show them pushed far out to the distant, empty edges of the tumor tissue.
It's like looking at a satellite heat map and seeing the actual front lines drawn in the tissue.
Yeah.
The human tissue perfectly mirrors the mechanics discovered in the lab. The builders lay the foundation, and the blockers are kept at bay.
It totally validates everything. It confirms that these aren't just, abstract laboratory phenomena. This chemical turf war between fibroblasts is the actual biological reality dictating patient survival in human pancreatic cancer.
That is incredible. So let's do a quick punchy recap to synthesize exactly what we've learned today, because the density of these mechanics is huge.
Good idea.
So first, pancreatic tumors hide behind a desmoplastic wall. They hijack their own structural scaffolding, using a jamming signal TGFβ to force local fibroblasts into becoming hostile blockers, or myCAFs.
Right. They pour the concrete and crush the roads.
Exactly. But second, the scaffolding is pliable. If we block that TGFβ jamming signal and hit the LTBR alarm, we can reprogram the tumor. We force the hostile fibroblasts to shed their identity and transform back into builders, or rCAFs.
Which then secrete the molecular Velcro and chemical breadcrumbs?
Right. Drawing B cells and T cells out of the bloodstream to construct permanent immune outposts, those pop-up bases, right inside the tumor.
We crack the concrete, build a base, and let the immune system do what it evolved to do.
It's brilliant, emphasizing the big takeaway from this cancer cell paper. It's that the next era of oncology isn't just about upgrading the weapons we give the immune system. We've spent a decade engineering better T cells. The future is about re-engineering the very architecture of the battlefield so those weapons can actually step foot on it.
I couldn't agree more. We have to manipulate the local environment to turn a cold, hostile tumor into a permissive battlefield. Because if we control the real estate, the immune system will follow.
Which leaves you with a wild, provocative concept to consider as we wrap up. We're talking about utilizing chemical signals to force a deadly tumor to spontaneously construct a highly complex, highly organized lymph node inside of itself. The local scaffolding of our bodies is remarkably obedient to the right chemical instructions. So if we can manipulate fibroblasts to build an immune base inside a pancreatic tumor, what other damaged or diseased tissues in the human body could we theoretically redesign just by hacking the chemical instructions of the local scaffolding?
It really makes you wonder.
It completely changes how you view the structural matrix holding you together. Thanks for joining us on this deep dive. We'll catch you on the next one.